Great. So I'm Aaron Simonds. I'm a sixth-year grad student in the lab, and I've had the good fortune of being involved in the CyTOF project for a long time, since the very beginning. And about a year and a half into our journey in playing around with the CyTOF, we met up with a computational biologist slash electrical engineer in the radiology department at Stanford, Peng Chu, who was working on an algorithm to look at high-dimensional data sets. And with the help of a statistician in our lab, Karen Sachs, they kind of transformed this algorithm into something that we could use to look at high-dimensional flow cytometry data in a more intuitive way. And I know it's kind of late in this seminar series for you guys to see this set of slides. I think in future years we should do this earlier, because I think SPADE has become so integral to the way we look at our data now that you've probably seen a dozen or 2 dozen SPADE trees by now without really getting a full explanation of what those things are. So I'm going to try and walk you through SPADE and how it works, and then a little bit of the, because this is a training course, the practical side of setting up your own SPADE analysis. So just a brief bit of background and the incentive for looking at the data this way. Of course, with traditional gating, we typically use biaxial plots. And as immunologists, what we tend to do is draw polygon gates around these plots and drill down into subsequent serial populations until we get narrowed down to the cell type of interest. And these gates are, by nature, only restricted to 2 dimensions. So you're only using the information from 2 dimensions at a time, and you draw these polygons. And often we have to just kind of cut off these continual relationships between the data. The other incentive is that there's just a lot of parameters in CyTOF data. So with a 4-parameter flow cytometer like a caliber, you could look at all of the pairwise relationships in your data using just 6 biaxial plots. And as time went on, cytometers got more advanced, and you need more pairs of plots, or more sets of plots, to look at all of the relationships between your data. Maxing out an LSR2 in our lab, you would have to look at 91 plots to look at all the pairwise relationships. But with a CyTOF with 31 parameters, you quickly reach around 500 biaxial plots. And we're now scraping the upper end of 40 parameters. So this problem is just getting worse and worse as time goes on. So obviously, biaxial plots are not a scalable solution. The third incentive here is that there are relationships in the data that we can probably take advantage of. And so I mentioned that there's continuous relationships that we like to segment with polygon gates. But this is from a review in 2004. There are lots of these beautiful sigmoidal relationships through the data, nonlinear relationships and surface marker expression patterns as cells differentiate. So this is bone marrow and 4 different differentiation pathways for B cells, granulocytes, monocytes, and erythrocytes. And you can see these continuous patterns and where the immunologist kind of broke these up into different groups of cell populations. So taking advantage of the information between these transitional populations, we use this algorithm SPADE, which again came out of a collaboration with Peng Chu and Sylvia Plevritis at Stanford. And the way this algorithm works is it looks at all of your data, the single cell data, as basically points in a cloud. And so this is a 2-dimensional cloud of data. And the denser regions of the cloud are these bright areas here on the plot. And there are really 3 abundant phenotypes and then 1 more rare phenotype. And everything's connected here in the middle. And the idea was that in a density downsampling step, you would try to normalize the density across this whole data set to make it more amenable to hierarchical clustering steps later on. And I'll show you why that works in a minute. So what we're trying to do is get rid of the overrepresented cells in these very abundant phenotypes while still maintaining the outline of the cloud. We also want to get rid of extreme outliers, like these couple of cells down here that are just noise. Those end up getting trimmed away as well. But we've maintained the shape of the distribution of cells in the data. Then, using a flavor of hierarchical clustering, this gets broken up into different neighborhoods, which we've got lots of names for, which are pretty entertaining. We could go through those afterward. But I've lately started calling them microphenotypes, because I think they are very subtle differences in the surface markers generally that we use to draw these trees. And so computationally they are phenotypes, although functionally you would have to use surrogate measures to figure out if they are functionally different cells. And then we just, for visualization purposes, we link together each of these different neighborhoods of cells as a node, a dot, along a tree structure, a branching tree structure, which uses the shortest or simplest path to connect all of the different nodes in the data set. And then going back to the original data set, remember we threw away some of the more abundant cells here. We bring those back in a step called upsampling, where they get assigned to the closest node on the tree. And then for visualization, we swell the size of those nodes. They get bigger as we add more cells back into them, just so you know that there are more cells in this node than this node. And then finally, we overlay the intensity of the different markers in each of those nodes. So this is the median intensity of marker 1 in that node on a blue to red color scale. And then when I switch to marker 2, you can see that marker 2 increases vertically across this structure, and the overall shape of the structure recapitulates what we had in the original data set. So in this very simple example, the data itself was only two-dimensional. So we can represent all of the data in 2 dimensions on the slide, and the tree looks just like the original data set. But the nice thing about this algorithm is that it's scalable to lots of dimensions. And so we can handle 30-dimensional data or 15-dimensional data very easily. And at the end of the day, what you will always end up with is a 2-dimensional representation of those relationships. And the way things are linked together on this tree is a clue to how close they are to each other in n-dimensional space. For ease of use, we basically rewrote the prototype of this program. Zach Bjornson and Michael Linderman in our lab rewrote it in a different language, R and C++, to not only make it much faster, it's about 20 times faster than the original prototype, but it's more user-friendly with this graphical interface here where you can select on a group of nodes like these 4 here and drill down to the primary data here. So this is freely available at, sorry, I meant to put the link here. This is at cytospade.org. And we'll probably be going through later today and tomorrow how to download that and install that on your own computers. And then just moving forward, you're not even going to have to download that. There's a major project underway. Again, Zach Bjornson is involved, and the folks at Cytobank are being very helpful in making this part of Cytobank itself. So you'll be able to directly export your data from a Cytobank experiment to a SPADE analysis, all in Cytobank, all in your web browser. And then you can actually interact with this tree, dragging nodes around and drilling down to primary data, all in the web browser. So you don't have to upload or download a bunch of files. So that's a work in progress. That's not quite ready for release yet, but it will be out soon. So now let me just go through a little bit of SPADE analyses so we can get a feel for how this approach works. This is from the paper, the publication on SPADE in 2011. And what this is Kenny Gibbs in the lab did I think it was a 9-color experiment, fluorescence experiment on mouse bone marrow. And he looked at a bunch of markers that are typically used for breaking out certainly T cells and B cells and subsets of T and B cells and monocytes, but also c-KIT to pick out more immature cells. And then plugging all of that data in a very unsupervised way into SPADE, it gave out a tree structure like this. So the 9 parameter data is now distilled down to 2 dimensions on the page here. And as we look at the overlays of colors of c-KIT, CD11B, so there's a monocyte marker, 4 and 8 on T cells, TCR beta on T cells, and B220 on B cells and the subset of dendritic cells, we can see how the pieces of this tree are really segmented into the different well-defined cell types. And so by drawing little bubbles around each of those cell types, Peng and Kenny labeled this tree into the different known cell types. Now then going backward and to kind of reconfirm that this would make some sense, Kenny went through manually and gated each of those same subsets in the data using traditional biaxial plots, and then overlaying his gating on that tree structure. Peng drew these figures where in black are the nodes that were well represented in Kenny's gates, and it really recapitulates where those structures were found on the data in a very unsupervised way by the SPADE algorithm. So it was just kind of a validation that at least it can do as well as the human gator in this case, although human gating is, of course, flawed and very subjective. Now, in a much bigger analysis, you've seen this plot from Sean's talk. and if you've looked at the science paper from last year. And this is human bone marrow analyzed with 13 markers. And when we took those 13 markers, again trying to get a very broad representation of the different cell types in bone marrow, we broke up bone marrow into about 280 nodes here. And all of the major cell subtypes were well represented. And by drilling through these trees and looking at all the different surface markers, I was able to draw these loops around the ones that were well recognizable. You know, naive CD4 T cells versus mature memory CD4 T cells, for example. And the neat thing is, because the algorithm does that downsampling step, but it's preserving the rare cell subtypes, we still captured very rare cell types like hematopoietic stem cells and pre-B cells and these dendritic cells here, which were extremely low abundance in the data, but they were represented on the tree. Yeah.
Do you have a sense of the abundance for hematopoietic stem cells? What's the, how much you have to be in order to get it to the node?
Yeah, in this data set, it was 0.2% of the gated cells. So there were about, I mean, we had a lot of cells. We had about 250,000 cells going into this analysis. So I think it was about 500 HSCs in that node. But we actually, what we did here was we had lots of data from the same patient. So we actually stacked 28 files, FCS files, from the same patient and ran the SPADE analysis on all of that together. So it was able to pull the phenotype information from about 8 million cells to pick out these extremely rare cell subtypes. And I think that helped a lot in getting a continuum that was connected by these very transitional cell types that made some sense. Now, I just should mention the continuum idea here is not an overt depiction of biology, right? You can't look at this tree and say that NK cells differentiate into mature T cells, which differentiate into naive T cells. That doesn't make any sense, and certainly that's not how you should interpret these trees, right? These are just an indication of the shortest path through your data, the most parsimonious connection between all of the different phenotypes in your data is how this plot is represented. So don't overinterpret it too much. Yep. Mm-hmm. Yeah, each node is a distinct set of, like quantitatively, the amount of the 13 different markers that were used to draw the tree. But they may be very subtle differences. And it's not, like I said in the beginning, it's not necessarily an indication that they're functionally different cells. It's kind of like if you took that blob and you sliced it up into a dozen little pieces, they may be different, but they may not be. When we look at the signaling, we do see some differences. Yeah?
So how do you decide how many markers and which ones to use?
Well, we used all of our surface markers, so that was an easy decision. But the choice of which surface markers to use was really driven in the experimental design. We actually did this experiment before SPADE existed, so we got really lucky that we chose a good set of markers. What we were trying to do with the 13 markers we chose was to capture all of the major immunological subsets and then also be able to drill down as deep as HSCs using the Weissman Labs definition of CMPs, MPPs, and HSCs. So we included all the markers necessary to do that, and we got lucky. SPADE is very good, though, of taking advantage of the information between those markers, especially these markers that are kind of continuous blobs. You can find these nice continuous patterns between these blobs. Let me go on, and I think some of the other questions will become clear. So as I flip through these trees, CD3 expression on the T cells is clearly higher than the rest of the tree, but CD4 and CD8 are mutually exclusive, like you would expect them to be immunologically. We captured the hematopoietic stem cells, like I said, which are 34 positive and quite rare. And we see all of the B cells, which have CD19, and then the subset of those, which had CD20. CD33 is on the whole myeloid arm here, and then 11B on the more mature cells. And then CD123 was expressed well on the plasmacytoid dendritic cells, and surprisingly on a subset of B cells, which I've bounced this idea off of some people. Nobody seems to know anything about this. But if you're a B-cell expert, please enlighten me. Okay, and then the neat thing, like I kind of touched on this, we actually stacked the data from 28 different FCS files here. So we can integrate the data from overlapping staining panels and overlapping data and calculate fold changes, and we can also just add complementary pieces of information using the same spade structure. So in this experiment from the science paper, we had 28 different stims. It was really 23 stims plus 5 replicates of the unstim. And then we had the 13 markers that were used for clustering and 18 phosphomarkers, which were not used for clustering, but these are the functional markers that we eventually overlay on the tree structure to look at the signaling behavior. One of the neat things we could do, though, is take a complementary surface-only staining panel, which had that same core set of 13 surface markers, and as long as we drew the tree using only the common markers, we could overlay the information from those 18 additional markers. And you could repeat this process, theoretically, with half a dozen or more different staining panels, as long as you're keeping the information from these guys constant. And this is a very nice, quick way, an intuitive way to come up with a tree structure that makes some sense and then get lots of complementary information from these other markers. These were very, very helpful, actually, in picking out all those different immunological subsets that we use to label the tree. If we didn't have the information from these markers, I don't think we could have put quite as many labels on that tree. Just as an example, we didn't have a proper NK marker in our data set. We didn't have CD56 or CD16, but we did have a unique combination of 45, 38, and 45 RA, which in the combination with all the other things that there's negative for, just happened to be very specific in terms of picking out these nodes, which then when we overlay the information for CD16, which was not used for clustering, we see a very tight expression pattern of CD16 on these nodes. And so that's how they got the name NK cells in our published data set. We also see 161 and CD7 on those cells as well, and CD56. So it was really a beautiful example of how SPADE can pick out information in the data that you didn't even know. Like, I never would have guessed that we could pick out those NK cells with the markers we had. The next thing you can do is look at fold change. So you can overlay the information with an additional surface marker, but you can also take advantage of the different expression patterns of your functional markers from condition to condition. Yeah?
So, we feed atomic data to SPADE to create these trees, and say that we also have data from an LSR experiment, traditional flow. Can we also feed that data to overlap?
I wouldn't really recommend trying to overlap those. You could try it. You would have to individually go through parameter by parameter and make sure that the scaling of the parameters is very, very similar and that you see the same distribution of expression with the two platforms. But that would be key.
Can we just take additional markers that are on LSR but not in cycle?
I'm sorry, can you repeat that?
Can we add markers instead of overlapping them?
I don't think so, if I'm interpreting your question correctly. But maybe I'm missing a point. Can we talk after? Okay, sorry. All right, and so in this case, what I'm trying to do is look at fold change between the functional markers. So if we look at just the median expression of phospho-STAT 5, which was not used for clustering, and we look at the median expression just like we've been looking at the median expression of everything else so far, we look at the basal state, we see a pretty even distribution across all of the different cell subsets. But then when we add in IL-7 stimulation for 15 minutes, we see activation in the T cells and NK T cells, indicated by the red and orange coloration over here on the right. Now, we can just overlay those two data sets and subtract one from the other, basically looking at fold change now. And using a blue to yellow color scale, we can indicate the activated nodes here, which are T cells and NK T cells, in response to IL-7. So this is nice immunological confirmation of the nodes that we call T cells and NK cells are functionally responding as those, but also, we're getting a very nice visualization technique to take a look at the signaling data. We see the same type of cell type specific responses to BCR cross-linking in B cells here, and LPS activation of P38 in monocytes, which is one of the things we did yesterday in the experiment. And then using another marker from that complementary surface panel, CD14, this is one of the LPS co-receptors in humans. This is nicely co-expressed on the monocytes, which are responding to LPS. So again, everything's kind of making sense immunologically here. Maybe this will help with your question about LSR2. So the example, the 9-parameter bone marrow example I showed was fluorescence data from mouse bone marrow. This is a even more complicated panel from mouse, what is this, 11 color, I think, from Drew Hotson in the lab. And he was looking at mice infected with salmonella and looking to see which cell subsets were expanding over time, over a time course. And I'm honestly not an expert at this, so I'm just going to kind of flip through and show you the slides that he gave me. But basically, as he flips through and looks at the different surface markers, he can pick out certainly T cells and B cells, granulocytes rather, monocytes, and dendritic cells here, and macrophages. And then when we look at a couple of activation markers, 44, 62L, Ly6C, and 49D, we can break this up into even more subsets. And then finally, Ki67 is a marker of cell cycle activation. There wasn't much going on here in the basal state. But then now here what we're looking at day 4, the coloration is no longer a surface marker. This is actually the change, the fold change in the percent of cells and where those cells were located on the tree. And we see at day 4 post-infection in the salmonella-infected mice, lots of granulocytes and macrophages were basically proliferating in the spleens of these mice. And then as time goes on, we see expansion in this compartment here, and then the 49D positive Ly6C negative cells, which I think is a subset of granulocytes, but please just jump in and correct me if I'm wrong. These are expanding rapidly at the later days in the time course. And then eventually it starts to subside, and we do see activation of some Ly6C positive T cells down here. And believe it or not, this is what Drew was looking for in the data. And he had found this after 2 months of digging through the data, the same exact data set, manually using biaxial plots. And he picked this out, and he was like, wow, this is really exciting. And when he finally plugged it into Spade, given he knew what he was looking for, but he found it that same day. So this can be a very enlightening approach to dig through your data. Again, because this is a training course, I want to touch on the different user configurable parameters. These are not in your slides. I apologize. I just made these slides last night. So I can give you a copy of these after the talks today. But basically, there are these default settings on the Spade software, whether you're using the online version or the offline version. And you can set the scaling factor, the amount you want to downsample the data, the number of nodes you want in your tree, and then a couple of things down here which are not actually that important. I'm going to skip over those until the end. So scaling. Scaling is certainly a very important thing here. This is a figure from the supplemental in the science paper, and it just shows you with a different ArcSign-H cofactor, which is what we call our scaling factor when we use ArcSign-H transformation of the data. With a high cofactor here of 150, fluorescence data, like LSR2 data here, looks very good. What it's doing is it's squishing the lower data near 0, and you're getting better resolution of the higher data on the plot, the higher intensities on the plot. But using the same cofactor, mass cytometry data looks very squished against the axes, almost as bad as it looks with the linear scaling. Using a lower cofactor of 5, mass cytometry data starts to look more like fluorescence data with the same parameters. And this is what we typically use for all of our CyTOF analysis. The reason here is that CyTOF data tends to be close to 0. 0 is the minimum measurement on our instrument, and because the sensitivity is not so great and because there's very little background, we are often dealing with data that's important data, and it's between counts of like 5 and 50. And so those low values here are important to us, and we want to capture them. But with that same scaling cofactor, fluorescence data looks really stretched out away from 0. And then with a cofactor of 1, neither of them looks very good. So this is, again, this is in supplemental figure 2, I believe, of the science paper. And it's worth looking at as just a cue to help pick out your ArcSign-H cofactor. And in Cytobank, you can easily tweak this factor and take a look at how it looks at your plot. In terms of Spade, what you really want to get to is a plot that makes some visual sense to you. Because Spade is going to be looking at these distances in ArcSign-H transformed space, just like you would be looking at them on the plot here. The next thing I want to touch on is density, because this matters when we're talking about the downsampling step. So very simply, I'm going to gloss over some specifics here, but the way that Spade thinks about density is it goes through cell by cell. And so for this red cell here, it looks within a certain radius at all of the number of neighbors that that cell has. And so in this case, the cell has 6 neighbors, so the density of this cell is 6. This cell only has 2 neighbors, so its density is 2. Now, as it does the downsampling step, it's probabilistic, and the cell with a higher density is more likely to be thrown away in the downsampling step than the cell with a lower density. And when you do this thousands of times through your data set, what you end up with is you're preserving the cells that have fewer neighbors. These are the more rare cell subtypes, while you're throwing away the T cells, for example, which have lots and lots of identical T cells right next to them in the data. So in the default of downsampling to 5,000 events, I'll show you what should ideally happen, and I'll leave it as an exercise to you to think about how this could go wrong if you didn't have 30,000 cells starting. So in the before example, your data might look like this. On a certain marker, you have the intensity going up here on the x-axis is just a regular histogram of cell counts, and you've got a more rare population and a very abundant population. And let's say this is 5% of your data over here. If we tell Spade to downsample to 5,000 cells, it's going to end up throwing away 25,000 cells in your data temporarily, but for the downsampling step. And so it's going to chew away at the top of this peak and a little bit at this peak as well, because these cells have a higher density. There's more of them with the same phenotype. And these are going to be thrown away with a much higher probability than these guys here. And at the end of the day, you should have exactly 5,000 cells with this distribution now, something like that, although it'd be smoother, to be honest. And now you've changed dramatically the relative representation of these cell phenotypes. This is now 80% of the data instead of 95%. And your rare cell subtypes, some of them got thrown away, but most of them are still there. And they're more represented in the data. So when you do clustering, these are more likely to form their own clusters in the Spade tree. That's the whole point. Like I said, as an exercise, try to imagine now how this could go wrong if you had 300,000 cells, let's say, and you still told it to downsample to 5,000 cells. Bad things can happen. Okay, now downsampling to 5th percentile. This is another possibility in the data, is to downsample to a percentile of the density. To be honest, I don't think this is the best feature to have in there. It's difficult to understand, and it doesn't always do what you want. But the way it's supposed to work is that if you look at the local densities of all your different cells, this isn't intensity anymore. The bottom 5th percentile, these guys that are more rare, what you're saying is you want to downsample the data until you've maintained the number of cells necessary to capture all the cells with that percentile density. So if you say to downsample to 5th percentile and that it's got a nice normal distribution here, that should be about 5,000 cells if you were starting with 100,000. And it'll downsample the data until you have 5,000 cells remaining. Now, with 13 parameters, this works out nicely, and you get this nice normal distribution in like PBMCs, for example. But I've seen it where with the same exact data set, if you do the clustering on 30 parameters, the local density is much more biased towards these cells that now have no neighbors. This is the curse of dimensionality, where in 30-dimensional space, cells are much less likely to have any neighbors. And so most of your cells have a very low density here. And as a result, the bottom 5th percentile is actually like half of your data. So you're not doing very much downsampling at all if you do it this way. So you really have to think about these things when you're doing it, and just play around. Just as you're tweaking these buttons on Spade, it doesn't hurt to run it over and over again. Just keep in mind that things like this can happen. The number of nodes is really something you should just play with depending on your data set. And the number of clustering samples you shouldn't touch. And the size scaling factor is just a visualization thing. This just tells you how big the nodes are. So this is my last slide. Basically, from my perspective, I think Spade, one of the nice things about it, and actually I think there are a lot of great tools on the forefront of coming out to look at this type of data, and they've got a lot of these different features, actually. You know, the problem of looking at high-dimensional flow data didn't exist before, so there are different groups, including some friends of ours and collaborators of ours at Columbia that are actually here in the audience, that are working on tools to do these types of things, and I think it's only going to get better and better. And Spade is just a milestone toward something that's truly robust and useful to everybody. But for now, what I like about Spade is that it is unsupervised. I think that's very good. It's good to take a broad look at your data and drill down to the things that matter to you. It looks at everything in all the dimensions, whereas by eye we can only look at 2 or maybe 3. And it displays rare cell subsets thanks to that downsampling step. And then the clusters have hard borders, which is easier for me to at least to understand instead of these probabilistic borders that other software like Gemstone uses. And then the caveats are, certainly, it's a visualization technique. Partly that's because it's stochastic, but basically just treat it as a way to look at your data. This is bad, though, because it's manual. You have to look, really stare at your trees with your eyeballs, and won't just give you the answer. So in some of our larger data sets, we've got 30,000 trees to look at, and that's not really practical either. So we're kind of back at the dimensionality problem. It's stochastic, meaning that the trees always look different because of a few of the steps in the algorithm. The data is slightly different every time you run the algorithm. And then choosing the number of nodes is done manually. And then we touched on this earlier. The number of nodes does not necessarily mean you have that many functionally distinct, immunologically different cells. So you have to interpret the data carefully in that respect. So that's it. I should definitely thank Rob Brugner, who's done a great job of stress testing the algorithm and attempting to improve it, although I think it's looking like there are even brighter things on the horizon. Michael Linderman and Zach Bjornson, who did an amazing job of converting it from a prototype into a usable everyday thing. And then, I'm sorry, I forgot to put Peng Chu and Sylvia Ploveritis on this slide, but they were obviously instrumental in coming up with us and being great collaborators to get this thing in our hands so we could use it early on. And thank you so much for your attention. Yep.
Post-clustering, can you get back to the raw data where you have CSV with the column annotation of which cluster it was in?
Yeah, actually, in the newer version of Spade, the one that's downloadable at cytospade.org, it outputs an FCS file, your original FCS file, and every cell is annotated in an additional column with the cluster they were assigned to. So you can actually play around with that in other programs like R. Yep. All right, if you have any questions after.